Welcome back. In the previous videos for APRICOT software, I have shown you how to install it, how to work with it. In this particular video, I'll show you what results are obtained, how the analysis creates the file. So which is right here, input and output. So I can quickly run it for you so you know what I'm talking about. So APRICOT creates the input folder called APRICOT analysis. It takes the input query, extracts information from UniProt. It takes our keyword of interest, extracts the domain from the database. It predicts the domain in our query proteins. It selects the proteins which have domain of our interest, scores them, classifies them, summarizes the result for the entire analysis, and formats them into the desirable format of HTML or Excel. So as we have defined already our analysis path as APRICOT analysis, it saves our input and output. I'm going to clear this up and yeah. So it has our input and output folder. Inside the input folder, there are three main folders: query proteins, UniProt reference table, and mapped query annotation. Query protein comprises of, I'm just gonna open the file. It's basically the query input that we provided, which are these two UniProt ID. So in my previous video, I said it's salmonella. It's basically not salmonella. It's E. coli proteins, but the name of the proteins are the same. This is E. coli HFQ, and this is E. coli ALAS protein. In the UniProt reference, you have a table which basically extracts all the information that you can obtain from UniProt table. Let's see what's in there. So it has entry, which is UniProt ID, entry name from the E. coli, its status, if it's reviewed or not, protein name, gene name, organism, how long is the protein, and if there is a gene ontology known for them. Another input folder that we have here are called mapped query annotation, fast path mapped query, mapped protein XML info, table XML path mapped query. So if we look at FASTA path, APRICOT downloads the FASTA file for our query proteins. If we look at XML path, we have XML files downloaded by APRICOT. The information from XML file are saved into this file called query feature table. We can quickly have a look at it. So it looks a bit messy because of a lot of gene ontology term right here. However, if we look at the header, we know what we extracted from the XML. So it's entry name, gene, locus tag, organism, PubMed ID, different reference for the cross-refs purpose, gene ontology, its existent type. So this was about all the input file, whereas user provided us, in this case I used two E. coli protein and we used the domain information. So if you want to look at where the domain informations are, it's basically in our source file domain data. So there are two sources, CDD, where we extracted KH related domain information, RRM related domain information, and these two are merged into all keyword selected CDD data. Similarly, for interpro, we have KH related interpro terms, RRM related interpro terms, and both of them merged. So if you notice, there is a file called, file called all keyword selected domain data, and if we look into it, These are all the selected RRM or KH related domain extracted from CDD and Interpro. Now what is in the output file? It's numbered by different subcommand that's run one after one. Here, this is for subcommand predict. Then you have compile information, you have selected domain information, annotation scoring, additional annotation, and analysis summary. The formatted files are in the format output data, and visualization files are stored in this folder. If we look at APRICOT analysis output, 0 predicted domain, so which is the first step. For both CDD and Interpro analysis, we have for each protein domain prediction. So if we look at CDD, the information are stored in these two files. If we look at IPR analysis, information is stored in these two files. Similarly, if we look at the compiled information, there are two folders, selected data and unfiltered data. In the unfiltered data, all the prediction by CDD and all the prediction by Interpro are saved, just so the user can go back and look into it. But in the selected data, only those are saved which are selected by our domain of interest. So for the CDD predicted domains, the data is stored in cddfilter.csv. This one is only ID for reference purpose if you don't want to look into everything, but just look at protein and which domain is predicted. And then for Interpro, you have it saved in iprfilter.csv. We can quickly have a look into that. So here is what you will find if you look into the selected data. You will have resource name, UniProt ID, resource ID for the domain, domain ID, which is the CDD or Interpro main ID, short name of the domain, full name of the domain, what domain keyword selected them, if there is a gene ontology known for it, if there are members from different database for the same domain, what's the length of the domain, start and stop site, e-value, bit score, coverage length, coverage percentage. So coverage length is basically how much of the reference domain is selected or identified in our query protein. Similarly, the identity, similarity, gap, and at the end, if it's selected by our parameter or not. So seems like the first one is selected by parameter, however the second one is discarded. Now we can look at different folders. Selected domain information. Here you will find classification of the predicted domain which are selected. Here is everything combined. As I said, that this is basically used for larger number of proteins. In our case, just combined is enough. So we can have a look at that. So here you have all the information. And as we assume this protein, which is HFQ from E. coli, has the domain of our interest, which also passes our filter parameter. So these are all the information that you will have for this protein, where the 40% of the coverage and 25% of the similarity are allowing this particular protein to pass the filter parameter. Quickly going to show you rest of the folders. If there is annotation scoring, they will be saved here. Additional annotation like localization, tertiary structure, and so on. Analysis summary. Quickly gonna have a look at analysis summary. Sorry. 5. So this is what it tells you: how many query proteins were there, how many were available in UniProt, um, if they are mapped with different IDs, in this case not, how many domains were selected for each term, how many proteins were selected with the domains from CDD and IntPro, which is down here, how many were total prediction, unique domains, unique parameter, domains that pass the filter. At the end, it lists only those protein which are selected with our domain of interest and they pass the parameter filter. So apparently nothing passed the parameter filter from InterPro. However, from CDT we could identify this protein, the HFQ from E. coli, that has domain of our interest. So this was about how you can browse through your analysis result. In the next video, we will go through the Docker information.